Okay, so in the last class, which was the first part of this chapter 6, we talked about enzymes. We talked about, particularly, we talked about transition state, um, how transition state is important in um, um, explaining to us how enzymes work. Transition state stabilization is the key factor that is important for um, uh, uh, catalysis, right? Enzyme catalysis. Um, so, okay. So we talked about also the difference between the free energy, standard free energy, the thermodynamics, and the activation free energy barrier. Remember delta G double dagger. I also tried to explain to you how certain reactions can have standard free energy positive and still may occur in the cell. And that's because actual free energy is sum of those two terms that includes standard free energy, right? We also talked about the mathematical model that actually is utilized extensively to explain the kinetics of enzymes. So that would be Michaelis-Menten kinetics. And Michaelis-Menten kinetics, we primarily talked about steady state kinetics, okay? And so in those, you must remember the Michaelis-Menten equation. You must know what individual terms mean, what are their units. You also know that Michaelis-Menten equations can be rewritten in the form of reciprocal plot. Remember double reciprocal plot, LB plot, Line-Weaver-Burk plot. Yeah, where you basically are plotting 1 by V versus 1 by S, right? So we talked about that, and then we ended up our discussion with brief introduction to enzyme inhibition. Enzyme inhibition that happens by a reversible mechanism. Today, we are going to focus our attention on molecular components of enzymes and different types of strategies that enzymes use for catalysis at the molecular level. Okay. So first enzyme that we are going to talk about is chymotrypsin. And chymotrypsin is a protease or a peptidase, and it's a serine endopeptidase, which means it basically utilizes a serine residue for catalysis, nucleophilic catalysis, and typically it cleaves the inside peptide bond, the bonds that will be found in the inside. It's an enzyme that actually is produced by the acinar cells of the pancreas, and it is a very important enzyme. And if you look here, we are showing on the left-hand side the primary structure of the fully active enzyme alpha-chymotrypsin. Okay, so it turns out that oftentimes all the proteases, and we have about 650 proteases in our genome. Proteases are stored inside your cells in the inactive form, okay, called zymogens. And that's because, you know, you want to make them active only when you want their function, right? You don't want them to be active all the time. Why? Because they might degrade your cellular proteins, which you might not want. So oftentimes, a lot of these proteases are stored in the form of what we call zymogens, okay? And they get actually activated by a very specific signaling cascades. And that's beautiful biochemistry that we'll not so much talk about. But this chymotrypsin is a protease that actually cleaves the peptide bond at the C terminus of the aromatic residues. So aromatic residues, three you know, tyrosine, phenylalanine, and tryptophan. So it actually will cleave the amide bond that exists at the C terminus of these aromatic residues. Remember, every amino acid in a peptide will have a C terminus and the N terminus. So the peptide bond that is at the C terminus is what chymotrypsin will cleave. Now, if you look at the mature structure of chymotrypsin, which is alpha chymotrypsin, you will see that it is comprised of three chains, A chains, B chains, and three chains. So there are three polypeptides with three amino terminus and three carboxy terminus. And you also, you will notice that there are some internal disulfide bond, intra-strand bond, and some that exist between the strands, right? And what they are showing you here is the three key residues, active site residues, which actually are important in catalysis. Those are the three showed here in the pink: serine 195, aspartate 102, and histidine 57. These are known as catalytic triad. Okay, these three residues are very, very important and actively participate in catalysis in the active site. There are surrounding residues that also play some roles in altering the pKs of some of these active site residues, but that's something that actually you definitely want to keep in mind. Cool. So the question is, as I said, these proteases are often stored in the form of zymogens, and how are they activated? Okay, it turns out that many of them are activated by proteolytic cleavage. Okay, so if you look at the chymotrypsin, the original polypeptide original peptide is a single chain peptide. Okay. So you can see that there is one amino terminus, and then there is one C terminus, and that's called, that's in a totally inactive form of chymotrypsin would be called chymotrypsinogen. That's fully inactive. It's one polypeptide. Okay. So as I said, it becomes activated after proteolysis by another protease called trypsin. Okay, so trypsin, what trypsin will do? Trypsin is another protease that will actually go and hydrolyze the amide bond at the C terminus of the basic residues. So either it has to be lysine and arginine, and you can see that trypsin will cleave at the 15th arginine residue at the C terminus. Okay, and thereby creating another form of chymotrypsin called pi-chymotrypsin. Pi-chymotrypsin is somewhat active. It's not fully active. It's a dipeptide. And it turns out that once you form this dipeptide, it is somewhat active. It will do autocatalysis. Okay, autocatalysis is pretty cool, right? It basically will chop its own part of the peptide. Okay, so what it does is it will basically chop away two dipeptides. Okay, so that will be serine 14 and arginine 15 and threonine 147 and acin 148. Those two dipeptides are actually cleaved. So now if you see, your dipeptide has become tripeptide. You can see that there are three C terminus and three N terminus residues, 1 to 13 as shown here in the left hand side, 16 to 146, that would be the B chain, and then you have 149 to 245, that would be the C chain. And if you remember, you pay attention to isoleucine 16 here, the amino terminus residue of the B chain. I will talk about that a little bit later on when I discuss some more fine details of chymotrypsin catalysis. So that's how you generate alpha chymotrypsin that is fully active. It turns out that trypsinogen is also stored in the inactive form, zymogen. And it actually is activated by what we call enteropeptidase, which will basically remove the 6 amino acids from the N terminus. So that's what they're showing you: valine, aspartate, 4 of them, and lysine. Okay, so active form of trypsin starts from the amino terminus, that is isoleucine, again at the 7th position, and 245 is the C terminus residue. That's the trypsin that is fully active. And again, trypsin, so with that, let's go to understand some more fine details of chymotrypsin mechanism. And to do that, I want to show you first crystal structure of chymotrypsin that is actually bound to one of its substrate analog. Okay. So we are seeing here, a close-up view of the active site, which has, the substrate analog bound to it. Part of that is, you can see in the green here. Okay. And the red part here is some of those key active site residues. The ones that are obviously seen here is serine 195 and histidine 57. Okay. And so you can see nicely that active site is, nicely formed, and you are seeing, active site residues that are important in this nucleophilic catalysis. So it gives you some idea how substrate binds. If you zoom in, right, you will see that's what is the active site looks like. Serine 195 side chain is shown here, serine hydroxyl group in the red, and they're also showing you histidine 57. The third residue, they're not showing you because it's buried, aspartate 102. What else they are showing you here? If you look here closely, you will see that there is this purple atom. That purple atom, you know what that is? That is the carbonyl oxygen. Okay. And so the carbon is right behind that in the green. And then this residue right here that is pointing at you is the NH, so nitrogen of the amide. Okay, so that's the bond that needs to be cleaved between the carbonyl and the amide. Okay, and you can see that that carbonyl oxygen is pointing towards these orange patches, right? These 2 orange patches. Those 2 orange patches are called oxyanion holes. Okay, oxyanion holes are conserved among many proteases. That's really cool how enzymes evolved, these proteases evolved to actually catalyze the reaction. Remember, amide bond is rock stable. You can actually leave a peptide and boil the peptide in water for umpteen hours and nothing is going to happen to the amide bond. It's a rock stable. But enzymes catalyze that very quickly because they use some tricks, and that's what we will talk about. Okay, so and here in the blue, not in the green, blue is the aromatic side chain. It can come from either tyrosine. I pardon my wrong spelling here. It should be TYR, not TRY. Or any of the aromatic side chain. You can see that there is a groove where these aromatic side chains go and bind. Okay. And the green part is the rest of the substrate. Okay, so that's how it looks like. So then people have extensively studied this enzyme, okay, and they know exactly at the molecular level how this enzyme catalyzes this peptide hydrolysis. Now, this is the mechanism, okay, as shown in details, and I will go over it, but you must go over it yourself after the class if you really want to understand. And you don't have to draw this mechanism with all the fine details. And I have another slides that I will show you how people actually write the mechanism of chymotrypsin if they're asked in the exam, because making all these grooves and coloring it is not really needed. Anyway, so The catalytic triad is shown here in the upper left corner. You can see that serine hydroxyl is shown here. They're only showing you the side chain, okay? Backbones, they're not showing. And then, so you can see that there is a histidine in the middle, and then you have aspartate. And what you'll notice is that there is a hydrogen bonding interactions between all of these three residues, the side chains, okay? So that's what is shown here on the leftmost side. Chymotrypsin, what they call is free enzyme, which will be E, right, in Michaelis-Menten. So chymotrypsin catalysis happens in two phases, okay? What we call acylation phase, okay, which is shown here on the top end, where you basically form this covalent acyl enzyme intermediate shown here on the extreme right. And when you form that covalent acyl enzyme intermediate, you have already cleaved the peptide bond that needs to be cleaved, which is shown here as product 1. You can see. But acyl enzyme intermediate, the enzyme is still bound to the other part of the substrate. Enzyme, remember, enzyme is a catalyst, so it must release itself so that it can take on another substrate and do catalysis. Catalysis is all, you know, you don't need stoichiometric amount of catalyst, right? Otherwise, catalyst will not be a catalyst. So it must reproduce. And so reproduction happens by the second phase, which is what we call deacylation step, okay, where you acyl enzyme intermediates gets broken down and you release the second part of the peptide that was remain bound in the acyl enzyme intermediate, and you regenerate the free enzyme form, okay? So that's how a full catalytic cycle of chymotrypsin happens. Now let's look at it at the individual molecular level. You can see that they are showing you a hydrophobic pocket. Remember, I told you that there is a pocket where the side chains, aromatic side chains will go and nicely bind. Yeah, that's what they are showing you. And then they are also showing you the amide nitrogen. Amide nitrogen, the backbone nitrogen of serine 195 and glycine 193. Those two amide nitrogen that you are seeing, those protons on the amide nitrogen, they are the oxyanion hole that are shown here in orange. And you can see that they are nicely pointing towards the carbonyl oxygen. So those are very important in catalysis. So what happens in the first step? Remember, Michaelis-Menten kinetics, substrate will go and bind to the free enzyme and form Michaelis-Menten complex, right? Or Michaelis complex. Non-covalent binding. If you look here, I always tell my students to pay attention and tell them also to draw some structures. Substrate is shown here as a polypeptide, right? And they actually want to cleave that bond right here between, you know, NH and CO. There's only one amide bond they're showing you here, right? In the center, right there. But the way they have written this polypeptide is reverse. Okay, so remember the N-terminal residue always be on the left-hand side, and C-terminus residue has to be always on the right-hand side. And so ideally they should flip this 180 degree, right? So you can see that how chymotrypsin is cleaving at the C-terminus of the aromatic residue. It's still the same, but they are just, you know, showing you differently here. I wish they could just flip it. But anyway, you know which polypeptide bond needs to be cleaved right there in the center. Okay, so they are showing you that thing goes and binds to the active site substrate. Now, when that happens, what happens next? I want to show you something that is again here. It turns out that serine hydroxyl, you know, its pK will be somewhere around 12, 15, let's say, you know. So it's not going to be, it's going to be protonated, right, around neutral pH. So this catalytic triad, what it does is it allows this alcohol to actually exist as an alkoxide. It actually enhances the nucleophilicity of this serine 195 oxygen. How that might happen? Think about it. If you have an aspartate which is negatively charged, if it is actually picks up that proton from imidazole, you can push these arrows and you can see that that other nitrogen, right? sp2 hybridized nitrogen right there is will become more basic. It will carry a net negative charge, right? And when you make that nitrogen more basic, it can actually pick up that proton from serine 195 and actually create a species like serine 195 in alkoxide form. So it enhances, this catalytic triad, the function of that is to enhance the nucleophilicity of serine 195 hydroxyl group. And that happens by increasing the basicity of that nitrogen with the assistance from aspartate 102. So what they're showing in the first step, that you can imagine that histidine 57 will pick up the proton. In that, you know that by now, that nitrogen is going to act as a general base, right? Because it's going to pick up a proton. So, and once it picks up the proton, serine O minus will be generated. O minus will attack that carbonyl, electrophilic carbonyl, and that double bond will get pushed. You will generate a negative charge on that oxygen. Now, negative charge of the oxygen, if not stabilized by something, this complex will fall back and you will go back. There will be no catalysis. Fortunately, because of the presence of the oxyanion holes, you'll be able to form this tetrahedral, a short-lived tetrahedral intermediate as shown here, right? After the second step, you can see that now that carbon has all single bonds. And that tetrahedral short-lived intermediate is stabilized by the oxyanion hole. So it does form. But then what happens is that enzyme undergoes structural changes, conformational changes, and then O minus pushes back, and then the other part of the peptide leaves. That becomes the leaving group. Remember, NH will be a very poor leaving group as shown here if it is not assisted by a general acid, right? So you can see they are showing you that that is assisted by the general acid, where histidine 57 is now acting as a general acid. It is going to provide the proton to the leaving group. So you actually produce the product 1 and you generate the acyl enzyme intermediate. Cool? Very cool, right? It's just the conformational change and you actually move forward along the reaction coordinate. The same histidine here was acting in the center, was acting as a general base. But once it picked up the proton, it was able to donate the same proton to the leaving group, which was the product 1. Pretty cool, right? Once you form the acyl enzyme intermediate, water molecule sneaks in. Conformational change happens and the water molecule sneaks in right next to that same histidine, histidine 57. And so in the deacylation step, beginning of the deacylation step, that nitrogen is again going to act as a general base, will pick up that proton from the water and make water a better nucleophile by making a hydroxide ion, OH minus. And that hydroxide ion will attack again that same carbonyl and you form this kind of short-lived tetrahedral intermediate in the deacylation step. Right? So again, you have same kind of tetrahedral intermediate that is being stabilized by the oxyanion hole. And then conformational change happens again and O minus collapses to form the double bond because carbon can have only 4 bonds. In this case, the leaving group is the serine hydroxyl of 195, right? Of the chymotrypsin. And that serine hydroxyl is again being assisted by the same histidine residue in the form of general acid. So it picks up a proton and then you form the product, the second product also dissociates and now you have formed the free enzyme. And this cycle happens, acylation and deacylation keeps on happening. It's a beautiful example by which you can see how one particular residue can act as a general acid, general base, both going forward to form the acyl enzyme complex, acyl enzyme intermediate, and in the deacylation steps as well. And how coordinations of individual amino acid side chains in the active site allows this chemistry to happen. Very beautiful. And so this is another way to, my students always ask whenever we give exams, how do you want me to draw the chymotrypsin mechanism? So I have drawn this in a most simplest form. You can follow this if you like. Okay, so that brings us to the next phase, some more kinetics. Can we do some enzyme kinetics experiment and prove that this acyl enzyme intermediate does form? So whenever scientists have to actually study an enzyme, typically what they do is they try to create an artificial substrate for the enzyme. Okay, what kind of artificial substrate? Substrates that when are converted to product by the enzyme, you can easily monitor them. Okay, you can monitor the progress of the reaction. It turns out that chymotrypsin can utilize para-nitrophenyl acetate as a substrate, and it can rapidly convert para-nitrophenyl acetate to para-nitrophenol and acetic acid. Okay, and you can see this is an ester, this is not an amide, and there are proteases that will catalyze both hydrolysis of an amide bond as well as an ester bonds. Not all of them, but there are plenty of them. So what scientists then did was, they basically took para-nitrophenyl acetate and they did what we call pre-steady state kinetics. So not the steady state kinetics. Remember in the steady state kinetics, the assumption, one of the assumption was that substrate concentration should be much higher than the enzyme concentration so that you can rapidly achieve steady state within the mixing time, within a second or so. When you are doing pre-steady state kinetics, what you do is your enzyme and substrate concentrations are typically comparable. So let's say you took 1 micromolar enzyme concentrations, you can have a substrate that is also 1 micromolar, or you can have a substrate that is 2 micromolar. Okay, so when you choose enzyme and substrate concentrations closer to each other, you basically will not be operating under steady state conditions. Okay, so that's what they did. So what they did was they basically took this para-nitrophenol in a buffer and added chymotrypsin. And this monitored the release of para-nitrophenol. It turns out that para-nitrophenol is yellow in color. Okay, para-nitrophenyl acetate is colorless. Once it forms the product is colorful, yellow in color. So you can easily monitor using spectrophotometry, right? So what they found out is that when they mixed these two together, they found a burst kinetics. Okay, burst kinetics are quite popular among enzymologists. And so you can see a rapid burst followed by a nice steady state rate. And so based on this observation, they concluded that the mechanism of chymotrypsin is consistent with a nucleophilic catalysis that involves the formation of an acyl enzyme intermediate. Okay, and then that acyl enzyme intermediate in the second step decomposes to give the free enzyme and the product. And so the reason they were able to conclude was that when you extrapolate this line to y-axis on the product axis, they noticed that the amount of product that was generated was the same amount that was utilized, same amount of enzyme that was utilized in these experiments. Okay, so if you used 1 nanomole of enzyme, you basically produced 1 nanomole in that burst phase. That's what it means. And so the way you can actually describe the mechanism is that you can draw a conclusion from this experiment is that there is a fast phase followed by a slow phase. Okay, two-step mechanism. And so what happens in the fast phase, enzyme will grab paranitrophenyl acetate and then form paranitrophenol rapidly. And that's why you are seeing if one equivalent of an enzyme is used, you will get one equivalent of paranitrophenol in the burst phase. And then if you have the extra nitrophenol still substrate, let's say additional equivalent, that will be catalyzed in the slow step here, in the second phase, which is the steady phase. And that second step is the slow step. So basically you form this acyl enzyme intermediate again. And in this case, you can see, and that decomposition of this acyl enzyme intermediate is the slow step. So because first phase is fast and second step is slow, you see this burst-like kinetics. And this is consistent with two-step mechanisms with an acyl enzyme intermediate. If it was a direct attack of water, okay, you can also catalyze this reaction in a very simple way, okay? You don't have to actually go through this thing, all this complicated step. All you need to do is bring the water molecule in, right? And activate that water molecule using general acid-base catalysis, and then attack, and the product leaves with an assistance from a general acid, right? One step. But that's not what happens in the case of chymotrypsin. So this kinetics is consistent with not a direct attack of water molecule. Instead, an acyl enzyme intermediate forms and a two-step catalysis is evident. All right. Now, scientists did some more experiments, okay? And I know that we are going a bit slow here, but let's pick up a little bit of pace. So they wanted to actually know what kind of residues are important in the active site and what roles they might be playing in catalysis, okay? So when scientists are actually trying to figure that out, what they do is they actually measure the rate of the enzyme catalyzed reaction at different pHs, okay? And so they do pH profile of the enzyme. In this experiment that you are seeing here on the left-hand side, what they did was they actually measured the rate of the chymotrypsin catalysis at different pH. But they carried out a reaction under kcat by km condition. This is important for you. Remember, the kcat by km condition is the phase where you have a linear dependence of substrate on rate, where the rate is proportional to the concentration of the substrate. Okay, when that happens, when the substrate concentration you choose is much less than the km value of the substrate, right? So if km value of the substrate is 10 micromolar, and if you're doing a reaction with the substrate concentration 2 micromolar, then you are operating under kcat by km condition. What is Vmax condition or kcat condition alone? Vmax condition is when you choose the substrate concentration much higher than the km value. So if the km value is 10 micromolar, if you choose to do the reaction at 100 micromolar of substrate, that means you are operating under Vmax condition, okay, where the rates don't change, substrate don't change. But in this experiment, regardless, they measured the initial rate at different pH under kcat by km condition, okay. So it turns out by using the methods of enzyme kinetics, this plot that you got can be broken down to its component, two components, kcat component and 1 by km component, using some mathematical manipulations. Okay. And when you do that, you get this plot B and C. And so you can clearly see that in B, the value of kcat increases from 6, 7, and then plateaus around 8. Okay. So kcat is like actually increasing as you increase the concentration. That's good for catalysis, right? So something must be getting deprotonated, right? You can literally think about this. At pH 6, kcat is low. pH 7, it is higher. So looks like more basic pH is liked by this enzyme. So some group must be getting deprotonated that is good for the catalysis, right? Obvious. What about C? C, you are seeing 1 by km plotted against pH. So 1 by km is decreasing, and it decreases after 8.5, pH of 8.5. And that suggests that 1 by km decreasing means km must be increasing. Km must be increasing means the affinity for the substrate must be decreasing, not good for catalysis, right? Okay, so there must be something going on around pH, after pH 8, where deprotonating maybe another group is really bad for the substrate binding, right? That's what you get from this plot. It turns out that scientists were able to go back and figure out what those residues are. Those two pK you see here, pH profile, those two residues that you are seeing the transition for in both kcat and 1 by km plot. Kcat 1 is for the histidine 57. That's the pK, that's the titration of histidine 57. So that makes sense. Why? Because that histidine 57, if it is actually deprotonated, then it will actually pick up the proton from serine 195, right? Yeah, so that makes sense. Oh, pretty cool. So that's actually histidine 57. That must be unprotonated. That could be the kcat, explanation for kcat. For 1 by km, they found out, based on some other studies as well, that was the alpha amino group of isoleucine 16. Remember I said don't forget about the isoleucine 16. That is the amino terminus residue of the B chain of chymotrypsin. It turns out that alpha amino group of chymotrypsin, isoleucine, if you deprotonate it, then it actually substrate binding is weakened. And so isoleucine 16 must be protonated for optimal activity. And people then went and looked at the crystal structure, and they found out that indeed, when you have the protonated form of alpha amino for isoleucine 16, it goes and forms a salt bridge interaction with another aspartate, not the aspartate, the catalytic triad, but aspartate 195. And that is important for catalysis. Okay, so that's how people figure out the mechanism. Cool. Now I want to show you something that you will appreciate, and that is that small structural changes can affect the kinetic parameters. What are the kinetic parameters, Michaelis-Menten parameters? Kcat and Km, or Kcat by Km, right? You just divide Kcat by Km. So these are Michaelis-Menten parameters, right? If people sometimes, people also call Vmax as Michaelis-Menten parameters, because Vmax is nothing but Kcat times total enzyme concentration. Anyway, so what you want to see here, they took 3 substrates, substrate A, B, and C. And they basically did the same Michaelis-Menten kinetics, steady state kinetics. They basically measured initial rate V at different substrate concentration, right? And they did the double reciprocal plot. They figured out what the kcat value was from that plot, what the km value was, and they tabulated it right here for you. That's all, very simple. One question that I often ask at this stage in this class, I ask my students to tell me which amide bond will be hydrolyzed in this substrate A. And majority of the students actually get this wrong. They end up telling me that the amide here in the left hand side is what will get hydrolyzed. And that's when they have not paid attention to what I have been telling. Because we are not able to ask questions because we are recording the lecture, I will explain that to you. Remember what I said. Chymotrypsin catalyzes the hydrolysis of amide bond at the C terminus, right, of the aromatic residues. So here is the phenylalanine, right? C terminus of phenylalanine is on the right hand side. N terminus of that phenylalanine is on the left hand side. So it's the right hand side amide bond that will get hydrolyzed. So the bond that will get hydrolyzed is CONH2 here. That's the bond, extreme right. Make sure you understand that. Anyway, so you can see that when they did that experiment, they measured kcat. Kcat was 0.06 per second. You have to pay attention to the unit. Km they measured was 31 millimolar for this substrate A. So they determined kcat by Km, that is 2 per molar per second. Units, pay attention to units. Then they took substrate B, where what they did was they modified this terminal amide. Okay, so they put this group here, right, acetamide group in pink. And then they went and measured the kcat and Km. They found out that their kcat doubled almost, right, 0.14. And Km also improved by twofold. Yeah. And so kcat by Km went up by fivefold. Specificity constant went up by fivefold. Small changes. So somehow you know that inclusion of that group in the pink improved both binding affinity, Km, as well as also improved the catalysis, chemistry part, the kcat value, right? When they took C substrate C, where actually they put a methyl group on that carbon, kcat improved even further. Look at this, right? 2.8. And Km became worse by twofold almost. But overall, kcat by Km improved by 114. Became 114. Dramatic improvements. So what I want to what I want you to take home is that small structural changes can affect kinetic parameters dramatically for different enzymes. And that makes sense. Enzymes are very picky, okay? Okay, with that, I want to actually talk about transition state analogs. Remember what I've been telling you. The enzymes are the best catalysts because they preferentially stabilize the transition state of the reaction, right? They don't preferentially stabilize so much substrates or products, but they preferentially stabilize the transition state. They lower the activation energy barrier of the rate limiting step, the difficult step. And so there is, and I have also in the last class showed you that stabilization is the binding energy of the transition state to the enzyme active site. So, you know, enzymes love their transition states, right? They bind transition states with much higher affinity. So, question is that if you want to develop inhibitors that will bind tightly to your enzyme and will inhibit the function of this enzyme, then you want to mimic the transition state, right? You want to design your chemical molecules that will more like mimic the transition state, not the substrate or not the product, but the transition state. And that is 100% true. Scientists have developed transition state analog inhibitors that are super, super potent, like affinity of 10 to the power minus 16 molar. Okay. I always give example of a very leading scientist who is right around the corner here at Albert Einstein College of Medicine, where I did my postdoc, Dr. Von Schramm, leading expert in designing transition state inhibitors, transition state analog inhibitors. And he does that by actually literally studying the transition states. Okay, and it's fascinating stuff. But anyway, so let's show you what the transition state analog inhibitors can look like. And sometimes you can actually use these transition state analog inhibitors to create artificial enzymes in the lab. Okay, so you can create an enzyme which will do a specific reaction. How? On the left hand side, I am showing you a reaction that is ester hydrolysis. Okay. On the right hand side, it's the carbonate hydrolysis. You can see the functional group carbonate and you can see the ester, right? Clearly. Don't just see it, you write it down yourself so you know. You don't have to write anything else. You can just say R1 or R2 on the other sides, but write the carbonate, write the ester, right? So when ester hydrolysis happens, you will see that hydroxyl group will try to attack that carbonyl, and your O will be, that carbonyl oxygen will actually, the double bond will get pushed. You will have delta minus charge on the oxygen, right? They're showing you partial bonds here, right? To show that that incoming nucleophile has partially formed the bond with that carbon, and carbon oxygen bond is no longer a strictly double bond. It has become a single bond with a delta minus charge on it. Remember, that's the one that gets stabilized by the oxygen and holes in chymotrypsin. So if you were to draw the transition state for that reaction, you can see that it has to be an atom in the center, like carbon, mimic, and 4 bonds to it, right? Tetrahedral bond, 1, 2, 3, and 4th one will be to the incoming nucleophile. Why? Because if you try to mimic that transition state shown here, see double dagger, they're showing you transition state. That's the sign that it is a transition state. If you can create a species that will have on this oxygen on the top delta minus and oxygen below delta minus, then you can mimic the transition state, and you might have a shot at creating an inhibitor that will bind to this enzyme that hydrolyzes the ester bond. So what will be that? So you keep those oxygen, right? And you, instead of carbon, you put a phosphorus. So this is a molecule called phosphonate ester. Why phosphonate? Because here phosphorus has 3 bonds to oxygen and 4th bond is to carbon. So phosphonate ester will be good inhibitor for the enzyme that catalyzes this ester hydrolysis, because you are trying to mimic the transition state right there, right? You can see that. What about carbonate hydrolysis? When you have carbonate hydrolysis, you can see that now you have carbon that has 4 single bonds to oxygen. So if you want to mimic that, you'll have to actually design a phosphate ester analog. So it's replace the carbon with the phosphorus, we keep those oxygens, and that can mimic the transition state of carbonate hydrolysis, and therefore this phosphate ester will bind to the enzyme that catalyzes carbonate. But vice versa, it will not bind. Okay, and so sometimes people use this kind of transition state analogs to create what we call catalytic antibodies that could actually catalyze a reaction. Okay, and so far luck has not been great. and people have gotten only 1,000-fold to 10,000-fold rate enhancement. But you can attach this molecule to a peptide, large peptide, create a molecular weight of more than 3,000, and maybe create antibodies and evolve those antibodies to do catalysis. So I already answered the question that is there in the bottom for you. So now let's talk about irreversible inhibition. And irreversible inhibition is an inhibition, type of inhibition where you cannot regain the enzyme activity after dialysis, infinite dialysis. It's covalently attached. Your inhibitor is covalently attached. The enzyme is completely dead forever. And so it turns out that a scientist developed this analog, diisopropyl fluorophosphate. You can see here, one of the oxygen has been replaced with the fluorine. And so when this compound was thrown to chymotrypsin, it turns out that enzyme lost the activity forever. Why? Because that serine hydroxyl group 195 went and attacked that phosphorus. This molecule was actually mimicking the transition state, tetrahedral transition state, and therefore got tricked into thinking that this could be the substrate. Actually, it turned out to be an inhibitor. This hydroxyl group attacked this phosphorus, and as soon as it attacks, the fluoride leaves as a leaving group. And then you have formed this very, very stable structure where enzyme is stuck. Okay. And then what you could do is scientists can go back and look at the residue that was covalently attached to the inhibitor, and they found out that it was indeed a serine 195. And that gave them a very good idea that it is indeed a serine 195 that is going to play a role of a nucleophilic catalyst in the reaction. Okay. So that was a very nice, clean experiment. I asked that question, but I will not ask that for now. Now let's quickly change gear and revisit before we talk about more mechanism. So what did you learn so far? So far, you actually learned how chymotrypsin, an endopeptidase that cleaves at the C-terminus of aromatic residues, does the catalysis, right, at the molecular level. You saw that some proof using structural studies, right, and enzyme kinetics. So that involved a nucleophilic catalysis, covalent nucleophilic catalysis. Okay, let's revisit. I wanted to show you one more cool proof that indeed induced fit is what most enzymes utilize for catalysis. Okay. So this is an enzyme basically called hexokinase. We'll talk about that in glycolysis. It takes beta D glucose, and the second substrate is ATP, magnesium, remember, cofactor. We talked about that, helps bind ATP at the right orientation where the gamma phosphate of the ATP can be transferred to the 6th hydroxyl group of beta D glucose, right? This one on the top. You can see they're forming glucose 6 phosphate. There are other hydroxyl groups, but hexokinase will only transfer the gamma phosphate to that 6th hydroxyl group. Pretty cool. Think about that. How specific that has to be. And so I will show you some example, but they actually have crystallized this enzyme when the substrate is not bound. So when you don't put glucose in the solution and crystallize the protein, hexokinase, what you will see is the protein that looks like on the left-hand side. It has a jaw, you can see active side, but which is open sort of. When you add glucose, or glucose analog or ATP analog, enzyme undergoes induced fit to accommodate the substrates. And so you can clearly see that jaw has closed on it nicely, right? So beautiful. Just think about it. Think about the breathing. Think about the coolness of this, right? So you can clearly see that upon binding of the substrate, glucose, hexokinase undergoes induced fit. Another very beautiful example. And it happens a lot. But scientists wanted always more and more evidence. They just don't settle down. So somebody did very clever experiment here. And I get very excited when we talk about this, okay? Instead of taking glucose, remember glucose, they're showing you open chain form, but open chain form doesn't exist to large extent in solution. It is in equilibrium with the closed chain form that you saw in this slide, right? Rapid equilibrium. But anyway, they're showing you just so you can appreciate here. They took xylose. Xylose, it turns out, is stereochemically similar to glucose. Okay, so if you look at this carbon next to that aldehyde, first one, you see same stereochemical configuration. Second one, same stereochemical configuration. Third one, same stereochemical configuration, right? But xylose is one carbon short. Okay, it has that CH2OH, but it doesn't have this one. So when they took hexokinase and fed xylose instead of glucose and ATP, what they were able to observe was that they didn't form xylose phosphate. No xylose phosphate formed. Very, very interesting. Think about it. There is no 6th hydroxyl group to form xylose phosphate. So it makes sense that xylose phosphate didn't form. But did it bind? Anything else happened in the active site? It turns out that xylose did bind to the active site. ATP did bind. But because xylose is one carbon short, ATP gamma phosphate could not be transferred to the 6th carbon, which is non-existent here in xylose. Instead, what happens is reaction started, catalysis started, and when the gamma phosphate bond was cleaved, it got transferred to the water molecule, H2O, instead of CH2OH of glucose. How did this notice that? Because it turns out that if you through xylose and ATP, think about it, and hexokinase, and nothing happens, if there is no binding, no induced feed, no closing in of the active site, creating a competent active site form for catalysis, then you will have xylose present, same concentration after, let's say, 10 minutes, and you will have ATP also concentration same, because it's lying there, it's not binding, nothing is happening. But it turns out that when they tried to look for ATP, they saw that ATP is rapidly getting hydrolyzed. And because hexokinase is not an ATPase, it doesn't hydrolyze ATP, that's not its function. The only explanation is that ATP and xylose bound to hexokinase in a catalytically competent conformational form. But because the 6th hydroxyl group was missing, gamma phosphate from ATP got transferred to water, and you were seeing hydrolysis of ATP. So enzymes gets tricked into phosphorylating the water molecule. Very cool, very cool. Now that brings me to talking about the second type of catalytic mechanism by enzymes called general acid-base mechanism. Not nucleophilic catalysis, but general acid-base mechanism. This is the first example I'm going to show you. Remember, in chymotrypsin, you had residues that were participating as a general acid and base. but it was an example of a nucleophilic covalent catalysis because a nucleophile was needed to do the reaction. In this case, like enolase, there is no nucleophilic catalysis. There is no nucleophile attacking anything. Instead, the whole thing is happening by general acid and base. Very, very cool. So enolase is an enzyme, glycolytic enzyme that catalyzes ninth step of the glycolysis. There are 10 steps. It catalyzes the ninth step. And what does it do? It basically takes 2-phosphoglycerate and converts it to phosphoenol pyruvate, PEP. Very important reaction. We'll talk more about that when we talk about. But this enzyme, it turns out, uses general acid-base mechanism. So if you look at the 2-phosphoglycerate structure on the left-hand side, you can see it's bound in the enzyme active site. And phosphoenol pyruvate is shown here on the extreme right. At this time, I will ask you to write down the structure of glycerol, okay, or glycerol phosphate, okay. But why? Because it will be important. We'll talk more about that later on. But for now, just watch this. So it is a 2-phosphoglycerate, right? At phosphate group, you can see at the second carbon. And you can see carboxylate at the glycerate. Glycerol, glycerate. Glycerate means carboxylate. The third carbon is oxidized fully. It turns out that active site of enolase, is very interesting. It has several positively charged residues in the active site. One of it is shown here, lysine. Okay, several basic residues. So one is lysine 3,4,5, and the other one is glue 2,1,1. Those two residues and 2 magnesium. These are the 4 key molecules that are going to play roles in catalysis. For binding, there are 2 magnesiums that are actually binding and stabilizing the negative charge on the carboxylate. You can see they're showing you diest interactions by dashed lines. When substrate goes and binds, it positions itself in such a way that lysine 3,4,5 is acting as a general base. Think about it for a moment. Typically at pH 7, lysine alone amino acid will be protonated, but not here in the active site of enolase. And as I alluded this to, because there are other basic residues surrounding this lysine 3,4,5 that actually lower the pK of this lysine. Therefore, it does exist as in the form of unprotonated form, NH2. So it picks up a proton right there. And then you push and you form this enolic intermediate. That enolic intermediate is nicely stabilized again by 2 magnesium ions, right? And then in the next step of catalysis, this O- pushes back this double bond. You have to do arrow pushing here. Double bond goes to form the double bond between carbon and carbon. And now the terminal carbon has 4 bonds because 1 double bond and 2 of these single bonds to hydrogen. This hydroxyl group must leave. But hydroxyl group, if it was not assisted by a general acid, will be a terrible leaving group and it will never leave. But if it is assisted by a general acid, such as here, glutamate 2,1,1, which is now existing in the protonated form, then the hydroxyl group can leave and you form water and you basically form the product phosphoenol pyruvate. Beautiful catalysis. There is no nucleophilic attack on any carbon, just general acid base, right? Very, very cool. And indeed, people actually have looked at the crystal structure of 2-phosphoglycerate bound to the enolase active site. They can clearly see that there are 2 magnesiums that are actually stabilizing the carbonyl oxygen. And they also see that the 2 residues, lysine 3,4,5 and glutamate, are there. Okay, but no covalent interactions between the substrate and those 2. Cool? Okay. Now this brings me to a very interesting enzyme. lysozyme. Catalysis of lysozyme is very much, fundamental in your biochem 1. Very well studied enzyme. Okay. It's an antimicrobial enzyme, that you find in various organisms, birds, plants, mammals, insects, bacteria, very well conserved. And it is also found, in your tears and saliva. Okay. So when your eyes are, your eyes are exposed to all kinds of things, right? Microorganisms, many bacteria. This lysozyme is there in your tears. I joke around with my students, biochem students. I tell them a little bit of crying is good for you, because when you shed tears, it has lysozyme. It will clear any infection that I might have. Yeah, but joke aside, it is a very, very interesting enzyme. Very small enzyme, 14.3 kilodalton. And what it does is that it catalyzes glycosidic linkage, beta-1,4 glycosidic linkage. Okay. So if you'll pay attention to here, and we'll talk about that, left-hand side, bottom left, I'm showing you the bacterial cell wall structure, okay, of a gram-negative bacteria. You can see there is outer membrane, there is inner membrane in the green, outer membrane in the orange, but then there is a single layer of this peptidoglycan, okay, this one in the blue, and you can see that they are tied together with these orange dashes they are showing you. And the integrity of that peptidoglycan is absolutely essential for bacterial survival. If you can inhibit any of those enzymes that actually form the peptidoglycan, you can kill bacteria. Okay, so you can create antibiotics. That's what it is. That's how antibiotics can be created, one way. So peptidoglycans, they must be maintained. So how does lysozyme kill bacteria? Basically, it goes and hydrolyzes one of the bond, glycosidic bond that connects these peptidoglycans. Okay, not to connect the two peptidoglycan, but actually that forms the peptidoglycan, the individual strand of blue. That would be the correct way to show. The peptidoglycan itself is a polymer that you are seeing here. It's made up of N-acetyl glucosamine and N-acetyl muramic acids. So they are showing you a polymer A, B, C, D, E, F. And this A, B, C, D, E, F is what the part of the peptidoglycan that binds to the lysozyme. Okay, on the right hand side, they are actually showing you a substrate bound to this enzyme. So what does this enzyme do? It hydrolyzes glycosidic bond between this D and E. Okay, so D is right there. So they are showing you in this circle here. You can see that muramic acid and glucosamine, right? That 1,4 glycosidic linkage. The lysozyme will catalyze hydrolysis. One thing you'll notice is that hydroxyl group, that water is adding from the top face of that sugar, muramic acid, N-acetyl muramic acid, right? So that is important. It doesn't come from the bottom. Stereochemical configuration of that carbon is retained. It comes from the top, from the phase at which the bonds are broken. Bond is broken, glycosidic bond. Important. So, people were fighting for the, figuring out how this enzyme works. So two mechanism was proposed, SN1 and SN2. And from your organic chemistry, SN1 mechanism is where you have a carbocation, generation of a carbocation. Carbocations are planar, sp2 hybridized carbon. And then the nucleophile can come from either sides and you can get a racemic mixture, right? That's one thing you form the carbocation in the first step, then it gets attacked by nucleophile and you form racemic mixture. SN2 type reaction is where the nucleophile comes from behind, backside attack, and you get the product with inversion of configuration, right? You know that. So in that context, we are talking about SN1 and SN2. So SN1 mechanism was proposed originally by Philip, is shown on the left-hand side. The SN2 mechanism is shown on the right-hand side, which is nowadays people think is more consistent with all kinds of data, structural data, kinetics data, isotope effect, and all that. Let's go through Philip's mechanism anyway to see how what he proposed. And SN1 type mechanism, what happens is that you can see there are two residues, one in the bottom of the substrate, aspartate 52, and the other one is glutamate 35, right? On the top face. And that's your substrate. That's the bond on the top has to be cleaved. So what he suggested was that that lone pair of oxygen from this N-acetylmuramic acid that will delocalize and form a double bond between oxygen and carbon. And therefore, oxygen will have a positive charge on it, but you can imagine that that positive charge can be delocalized between the oxygen and carbon, right? So that's why they are showing you partial. That's your carbocation, what we call glycosyl carbocation intermediate. That's what he suggested happens. When this happens, you can already see that the second part of the molecule has left. The bond has been cleaved already. How is that bond cleaved? When you made that double bond between that carbon and oxygen with a positive charge on it, that carbon of N-acetylmuramic acid cannot have more than 4 bonds, right? So that bond between carbon and oxygen, the glycosidic bond, that must break. And it cannot break because the leaving group is strong base. It gets broken because of a general acid assistance from glutamate 35. So that's how the leaving group leaves. So you form the first product, right? And you form this glycosyl carbocation intermediate. In the second step, what happens is that water molecule comes from the top phase. Because remember, enzymes are chiral. They have all these side chains. They can block the axis of substrate molecules from either side, whatever they want, however they evolved, right? So it turns out that water molecule will actually have access to this glycosyl carbocation only from the top phase, and therefore sneaks in. That glutamate 35 will then activate that water molecule, will pick up that proton, and that hydroxide ion will attack that carbon from the top phase. And so you form the second product, an enzyme has been released. So you started with the top phase, right? And you actually ended up with the top phase. So that makes sense. So people accepted this for longest time. You are able to see retention of configuration at that carbon of N-acetylmuramic acid, even when you go through the SN1 mechanism. But what about SN2 mechanism, which is more consistent with the data that we have latest? So that's really what's happening in real sense, is that there is a nucleophilic covalent catalysis. Yes, it's not just general acid-base catalysis. It's covalent nucleophilic catalysis happens. How? It turns out that aspartate 52 acts as a nucleophile, that carboxylate O minus attacks. This leaving group leaves with the general acid assistance, you form the first product, right? But you have this covalent intermediate, just like covalent intermediate you saw in chymotrypsin. In chymotrypsin, you saw with the serine 195, here you are seeing with aspartate carboxylate of aspartate 52 carboxylate. Then what happens? Once you have the covalent intermediate, the water molecule sneaks in from the top, activates the water molecule, water attacks that carbon from the top, leaving group leaves. In this case, leaving group is the bond between carbon and oxygen, the aspartate 52. And you have still the same product with the retention of configuration, and enzyme is back to its native state, free enzyme form, right? So it's covalent nucleophilic catalysis that is more consistent. although it is a SN2 type of a reaction, if you do SN2 type of a reaction twice, you will get retention, right? So it's possible. So that's, you can see here how you can still do SN2 and still retain the configuration of the leaving group. Cool, right? Very cool. And indeed, they were able to actually see in the crystal structure. What they did was they actually crystallized a substrate analog with lysozyme, and they were able to clearly see a covalent bond formed between the aspartate 52 and that carbon. That was, that scientist really rested the case forever. Problem solved, mechanism resolved. And so that was a really, really cool structure actually, and cleared up a lot of. Now that brings me to talking about another very interesting enzyme that utilizes covalent nucleophilic catalysis, like that of chymotrypsin and lysozyme, the latest mechanism. This is a bacterial enzyme. We are talking about bacteria. So as I said, you can see here, there is a lower membrane. The peptidoglycan is shown here in the blue polymer of N-acetylmuramic acid and N-acetylglucosamine. And they are stranded together by this orange line. That orange is the bond that I'm going to show you. Transpeptidation reaction, they call it. Okay. So, how do bacteria build this cell wall? How do they do that? It turns out there are many steps. Obviously, it's a whole biochemical pathways, but we are only talking about a transpeptidation reaction, which actually creates this mesh-like structure that gives additional stability to the cell wall of the bacteria. So you can see here, they're showing you sugar polymer in red and green. And then from there hanging is a peptide, you can see. And you might notice that there are some D amino acids as well. So as I said, nature has some D amino acids it utilizes, particularly bacteria. So how do bacteria build make this transpeptidation reaction? How do bacteria make this cross-linked peptidoglycan? By this reaction right here. The enzyme transpeptidase has a nucleophilic residue serine hydroxyl, just like chymotrypsin. Will attack a carbonyl. That leaving group you can see is D-alanine. Leaving group leaves D-alanine, you can see they're showing you. Then enzyme has formed this acyl enzyme intermediate, just like in chymotrypsin. And then amino group from the neighboring peptidoglycan chain 2, will attack that carbonyl and form an amide bond, rock stable bond. And in that case, releasing the enzyme transpeptidase. And so you have formed this nice cross link between the two peptidoglycans, and that cross link is the orange line here. Very, very important. If you can figure out a pathway to inhibit this transpeptidase enzyme, you can actually kill bacteria because of sheer osmotic pressure. They will burst open, die. It turns out that Alexander Fleming, that's exactly what he discovered. Okay. So Alexander Fleming, I don't know, I don't want to tell you the story because we're running out of time, but basically he isolated penicillin from a mold, okay, rare strain of Penicillium notatum. And the way he discovered that this mold was growing in a petri dish, he went on a vacation. When he came back, and this, he was growing a colony of staph, which gives you sore throat and infections and all that. Staph aureus bacteria. And when he came back after a vacation, he saw that, his petri dish, there was a mold growing, not bacterial colony. And around that mold, he saw that it was completely cleared, wiped out. So he figured out that that mold might be secreting some kind of compounds that must be killing these bacteria. And that's how penicillin was discovered. Amazing, amazing. So penicillin G that is shown here, penicillin structure is shown here. You can see there is a side chain and there is a beta lactam ring, what I call business end of the molecule, very sterically constrained beta lactam ring, covalently modified to the 5-membered thiazolidine ring right here. So that's the basic structure of penicillin. So Pen-G was the first one to be discovered, but it turns out that it is not really stable. It gets degraded by stomach acid, and therefore it should be injected by, injection. But then scientists have created hundreds of variants of these, amoxicillin, penicillin V and all that. Penicillin V, for example, is acid stable and it can be administered orally, And so what happened was, what the target was for the penicillin that he discovered? It turns out that the transpeptidase enzymes of bacteria, also known as DD peptidase, they were the target of this enzyme, this molecule penicillin. So how will transpeptidase get inhibited? Remember, transpeptidases have serine hydroxyl group. I just showed you. As soon as penicillin you throw at those bacteria, transpeptidase will think that it is a substrate. And I can't go into that because this is my PhD work. I can go on and on this, but don't want to. Just don't have time. Serine hydroxyl attacks that carbonyl oxygen and ring opens up. And you form this stable acyl enzyme intermediate. It doesn't deacylate. This enzyme is tied up with the penicillin. It cannot cross-link two peptidoglycans, and therefore bacteria die. Okay, that's how penicillin works. It will form the acyl enzyme intermediate with transpeptidases, and that's it, done. Bacteria eventually will die. It turns out that bacteria are very smart. They are evolving constantly. So they figured out a way to actually take care of penicillin. They became resistant to penicillin. You must be hearing a lot about penicillin resistance, antibiotic resistance. And there are more than 1,000 types of these other enzymes called beta lactamases. Okay, beta lactamases are these resistant enzymes that have evolved from transpeptidases, and they can take penicillin and completely degrade it before penicillin reach its target, which would be transpeptidases of bacteria. And that's how bacteria will keep on growing. So what happens to beta lactamases? What do they do? How are they resistance enzyme? They will do very similar chemistry as serine, transpeptidases. They will attack beta lactam ring, open up, but they have evolved to catalyze deacylation step rapidly. They can bring in a water molecule and completely turn over that acyl enzyme intermediate, and thereby breaking down the penicillin ring, and penicillin can never reach the transpeptidase target. And that's why bacteria become resistant. We have thousands strains of beta lactamases. Now, constantly we are under threat that all of our antibiotics will eventually get resistant. So people, what they are doing, and what my PhD thesis was, develop inhibitors of beta lactamases. So we can stop the function of beta lactamases, then bacterial resistance can be overcome. And so there is one beta lactamase inhibitor that actually is utilized in the clinic. Tomorrow you become doctor, you will know this. That is a suicide inhibitor of beta lactamases, which means this molecule will go and kill beta lactamases. And so it can prevent resistance. With that, let me talk a little bit about allostery. What is allostery? You already know this, right? In many enzymes, you have a catalytic unit, and then distant from the catalytic active site, you have regulatory unit. Okay, R represented, modulatory unit, where a positive modulator or a negative modulator can bind and affect the catalysis in the active site. The allostery means binding of one ligand at one site will influence the binding at a distant site. Through how that happens? Through conformational change, those things are communicated to the other side. So what you are seeing here is that you have a regulatory domain where a positive modulator will bind, right? You have a less active enzyme. When positive modulator will bind, it will convey conformational change in the catalytic unit. So now you can see that catalytic unit has become complementary to the substrate. Earlier substrate couldn't bind. So now you have a more active form of enzyme. Catalysis will happen because you can now form active enzyme substrate complex, right? So that's what allosteric is. You know that already. We talked about hemoglobin. I wanted to show you an example how beautifully this is done in our cells. There is an enzyme called aspartate transcarbamylase. This is an enzyme that is needed in the pyrimidine biosynthesis pathway. Okay, this is an enzyme that is made up of 2 trimers of the catalytic subunit, okay, each of 32 kilodalton approximately, and 3 dimers of regulatory subunits. Okay, so if you look here, 3 catalytic subunits are shown in the shades of blue and purple. So you can see here, blue and purple. You can see both phases they are showing you. And regulatory domains are shown here in red and yellow. So you can see regulatory domains interacting on the outer, right? On the left hand side if you look. So catalytic units are in the center sort of, and regulatory domains are out in the outskirt. Think how beautifully this enzyme has to do its job. Why this has to be so massive? It has to be so massive, and it has to have all these different regulatory domain, because the reaction must be very carefully controlled in metabolic pathways. Okay, so it just gives you an idea that to achieve optimum amount of catalysis in our metabolic pathways, how enzymes utilize structures, and sometimes very, very complex structures like this. Cool, right? And so what type of enzyme kinetics you might see with these allosteric enzymes, right? You may have question. This is catalysis. This is not just binding of ligand to a protein, like as was the case in hemoglobin to oxygen. This is catalysis. Most catalysts follow Michaelis-Menten kinetics, right? But if it is an allosteric enzyme, typically if you measure initial rate on the y-axis, right, at different substrate concentration, V naught versus S. This is not the progress curve. X-axis is not time. I want to emphasize again and again. I know I'm boring you with this, really stressing this out, stressing myself out with this. But this is very, very important. It's V naught versus S, Michaelis-Menten kinetics, right? If it's an allosteric enzyme, typically you will see the curve that you are seeing in the middle here when there are no modulators present. You see a lag sigmoidal type of curve. You can see a lag in the beginning, and then, V naught saturates. This is type 1 allostery observed for enzymes. Okay, at the bottom it says. Type 1, what does that even mean, right? I want to explain to you. When you have a positive modulator, you can see that your curve shifts to the left in presence of a positive modulator. That makes sense, because if the curve V naught versus S shifts to the left, you know that binding affinity is increasing, right? So that makes sense. If you have a positive modulator, the binding affinity for the substrate is increased, therefore the curve is moving to the left. If it's a negative regulator of this kind, type 1, then binding affinity decreases, and you see the curve that is shown here on the extreme right. What you will notice here is that all three curves are actually going to eventually approach and touch each other. That's how the curve shape looks like. See, you can only do experiments up to certain substrate concentration. You can't have infinite substrate concentration, right? But based on the shape of the curve, you can fit your data. You can see whether your curves are ever going to converge or near converge. And if you look at the plot like this, you can see that, yes, they will eventually converge because they are going in the right direction. They will all reach their Vmax. That's what I'm trying to get at. What that means is that type 1 allosteric enzymes will have the same Vmax. They will be able to achieve same Vmax provided you give them enough substrate concentration. But their Km is different. Okay, how is that evident here? Same plot. So Vmax is here, right? On the x-axis, y-axis, you can see it's crossing there. There is only one Vmax in all three cases. Half of that Vmax is shown by the line that is shown in the middle, right here on the y-axis, right? That's half of Vmax. Let's draw a parallel line to x and see where that line cuts each of these individual curves. Wherever it cuts, if you extrapolate that now onto the x-axis, which means you draw a line now parallel to the y-axis, you will get individual Km values. In this case, they are writing K 0.5. Remember, you can write K 0.5, you can write P50, you can write Km. All those are, similar. KD sometimes. So that's, they call it Km or K 0.5, right? So you can see that for each of the individual enzymes, they have different Km values, but the same Vmax. This is type 1. Km K 0.5 is altered without a change in Vmax. What happens in the type 2, which is less commonly observed in nature? Here, the Vmax is altered and K half is nearly constant. How? Look at the curve here. You can see that even if you try to extrapolate this curve to infinity, the middle curve is never going to approach the top curve, right? It's just not. So in this case, you have a Vmax, three types of Vmax: when you don't have any modulator, when you have a positive modulator, and when you have a negative modulator. In all of the cases, you have three Vmaxes, and you can see that they are actually showing you by where they are on the y-axis. Again, so if you try to take half of Vmax of each individual curve, right? And try to draw that line on the x-axis, you will see that they all go to the same point on the x-axis. And so K 0.5 remains unchanged and Vmax is changed in type 2 allosteric modulations, as you can see. That brings me to the last slide for this chapter, and which is about feedback inhibition. Okay, we talk about metabolism, but I want you to get ready for it. Okay, so that's why we talk about this inhibition right now. It turns out that we cannot make L-isoleucine. Okay, so we must obtain L-isoleucine in our diet. But plants and bacteria, they can synthesize L-isoleucine from L-threonine. There is biosynthetic pathways that exist. Cool? And the way L-isoleucine is synthesized in bacteria and plant is by these 5 steps, and each of those 5 steps have this enzyme E1, E2, E3, E4, E5, and they all grow through these intermediate compounds A, B, C, D, and then eventually you will get L-isoleucine. Cool? So when bacteria are trying to synthesize L-isoleucine for their need, right? So they will turn on this pathway and they will start making L-isoleucine from L-threonine. But after a while, they may not need L-isoleucine. And they may not need L-isoleucine in whatever protein they are trying to make or whatever they are trying to do with that isoleucine. The concentration of L-isoleucine will rise in the system. And once concentration of L-isoleucine rises because it is not being utilized, it turns out that it will go and bind to the first enzyme in the pathway, which in this case is threonine dehydratase, E1. Threonine dehydratase is the first enzyme. L-isoleucine will have a binding pocket, regulatory pocket on threonine dehydratase. It will go and bind, and once it goes and binds, it will block the activity of threonine dehydratase. Think about regulation, regulatory domain, binding a ligand here, L-isoleucine, and basically blocking the function of E1 enzyme that catalyzes the reaction that goes from L-threonine to A. And therefore it will shut down the production of isoleucine. This is what we call feedback inhibition. The last product of the metabolic pathway goes and inhibits the first enzyme of the pathway by feedback mechanism, but not any of the other 4 intermediates, A to D. Okay, that's pretty cool, right? That's where the chapter ends. And I have actually added, I have changed your Blackboard slides, I think. Added some more stuff. So whenever you see a change in something, you should go and download latest. And I have a couple of minutes, so I'm just gonna do few, jog your memory a little bit, okay? So please don't mind. I know your mind is occupied by the coming exam. This is going to be part of the second exam, but I want to just ask you some question, see how much you're absorbing. So this is on Michaelis-Menten equation, right? And there will certainly be a question in the exam, second exam, not this exam, please. Somebody did this reaction where they took the substrate, different substrate concentrations, and they measured initial rate. They measured the initial rate in presence of an inhibitor, which is on the right-hand side, and in the absence of the inhibitor, in the one that is shown here in the middle column. So if it was no inhibitor reaction, it will be simple S versus V, right? First column and second column. But they also were able to measure the initial rate in presence of inhibitor, different concentrations of inhibitor, right? And that's the rate they measured. So S versus V. So the question is, what is the V max in the absence of inhibitor? How do you get that? I'm just going to let you think for 20 seconds, okay? And then come back and explain to you. V max in the absence of. So you know that when you have to look in the absence of inhibitor, you are not going to look at the column that shows plus inhibitor, right? You ignore that.
100.
So you actually look at the S versus minus inhibitor column, and you will see that as the substrate concentration is increased to 0.2 unit, initial rate is 100. When it's 0.1 unit, still 100. 0.05, still 100. Oh, that looks like a Vmax rate right there, because initial rate doesn't change even when you double or quadruple the substrate concentration. So that rate is the Vmax, right? So what will be the answer? 100 is the Vmax in the absence of inhibitor, right? Okay, what is the Km in the absence of inhibitor? How do you calculate Km by looking at this table?
0.02.
Yeah, so don't speak because let me explain. I want to, because this is a recording, so we want to, I give you some time and then I want to explain to you, all of you. Km, you remember one way to determine Km is when you have V versus S plot, right? You see where the V max is, half of that V max you go, and then you draw a line and see where it intersects the curve, the hyperbolic curve. And we are talking about a hyperbolic nonlinear equation of Michaelis-Menten. And then you draw a line parallel to the y-axis and see where it cuts on the x-axis. That becomes the Km. Km is nothing but the substrate concentration at which V max is half. So Km in the absence of inhibitor will be 100 is the V max. So where I will look, where is half of that 100? Oh, keep going. I see here 50 right there. Half of V max is right there. So then what is the substrate concentration at that half of V max? That turns out to be 0.0002. So therefore the Km is 0.0002 unit, right? And so that kind of things you can determine by actually if you are given a table, right? So you already know Vmax, somebody gives you enzyme concentration, you can use the Michaelis-Menten parameters equations to determine some of the other parameters. So I want you to always make sure you practice. I have also uploaded some problem sets so you can do how they're solving, right? And that's where I will stop. So let me stop sharing, and then stop recording.